We've all worked hard today already, so this is the last talk, and I will try and keep it short and simple. So our eminent host published in 1996 an important paper which has been cited many times already today, and there it was stated that as soon as the temperature passes over 30 degrees, that this means heat stress. And when you look in the literature, and we've heard it several times already today, it's obvious that there's a difference between acute or even repeated acute heat stress versus chronic heat stress. And it makes a lot of difference. And I think it's also relevant to different conditions in different countries. I think in Europe we have spells of heat stress, but they're short, only during short period of the day, whereas in other conditions, of course, this is more like chronic and of long duration. And so I actually, when going through the literature, of course, I came up with the same sort of data which have been shared with you already several times today. And these data, they come from different papers. And this sort of sums up the most important observations that were made in the literature. And a lot of them are directly related to intestinal health. So I would like to focus on a few of these with respect to what they mean when we're looking at the health of the intestinal tract in the animal. And one of them is definitely the reduced antioxidant capacity. And of course that brings us to the previous presentation. We all know that antioxidant capacity and oxidative stress is very important for muscle. But we sometimes forget that actually the most metabolically active cells in the body are the epithelial cells of the intestinal tract. So if there's a cell in the body that is very susceptible to oxidative stress, it's the intestinal epithelial cell. And of course we know that the protection against this oxidative stress is mediated by a number of mechanisms that are incorporated in the cellular metabolism. And one of these metabolic pathways is of course related to the superoxide dismutase, and this superoxide dismutase it's key in the protection of the cells against oxidative stress. And of course we know that in the intestine, when there's oxidative stress and there's damage to membranes, this will inevitably lead to inflammation. And so inflammation is one aspect which is directly related to heat stress, inflammation in the intestinal tract. Luckily for us, the intestinal tract has been, coping with inflammation for other reasons as well. So this is just one trigger of inflammation on top of all the other triggers that are already there. But that is also probably the reason why under hot tropical conditions, when you look at issues like dysbiosis and intestinal stress, they seem to be much more severe. And probably this is the reason why we are in Europe, have maybe an easier life in trying to cope with dysbiosis and trying to cope with other stresses to the intestinal tract, because in tropical heat stress conditions, you have this additional pro-inflammatory factor that comes on top of all the rest. So in the intestinal tract, there are a number of feedback mechanisms which sort of dampen down inflammation. And I think we should keep this in mind. Heat stress is just one of the different stress factors that affects the intestinal epithelial cells. And so the way we handle them is to some extent similar as the way we handle other stressors of the intestinal epithelial cells. And that is also, for instance, through supporting the dampening down mechanisms, so the negative feedback mechanisms on the intestinal inflammation. And one of these dampening down mechanism pathways is the PPAR gamma pathway, which reduces intestinal inflammation through suppression of the NF-kappa B pro-inflammatory pathway. And this is just a simple practical list of known feed additives, feed ingredients that can act on the PPAR gamma pathway, that have documented effects supporting the PPAR gamma pathway, and thus can dampen down inflammation in the intestinal tract. And one of them I want to stress because it'll come back in some more slides, and I also want to show you, draw your attention to this one, to the glutamine, which was mentioned earlier on by Dr. Michael Kidd as an important factor. So is indeed something that seems to be quite universal. Then there's the aspect of heat stress increasing susceptibility to disease. And we actually worked together with a group in Greece some time ago in using our model of necrotic enteritis and using repeated acute heat stress. We could show that indeed the animals become more susceptible to necrotic enteritis. This is just numbers of birds that have almost no lesion or extremely high lesion, and you can see the red bars show you that there's more birds with high lesion score in the necrotic enteritis model in birds that have been repeatedly exposed to heat stress, which means that under heat stress conditions, necrotic enteritis might be more severe. So indeed, heat stress is increasing susceptibility to disease. But one of the most fascinating aspects, I think, is that heat stress is known to increase intestinal permeability. So it induces gut leakage, as we were told this morning already. And this is not something which is unique to the chicken, because there's a lot of interest in human medicine looking at athletes. You know, athletes doing extreme efforts, body temperature rises due to the efforts, and then you also see increased intestinal permeability. Now, gut leakage due to heat stress, and then you come to think of gut leakage can also be induced by, say, starvation. If you starve the birds for even only 22 hours, you already increase the permeability of the intestinal mucosa. So here again, heat stress comes on top of different other stressors at the level of the intestinal epithelial cell. And so that brings me to my favorite topic, and I apologize for those who have heard me before because I tend to repeat myself, but it's very fascinating to see that what is happening actually in case of heat stress is the tight junctions between the epithelial cells are damaged for the simple reason that due to oxidative stress, the epithelial cells have a hard time of producing enough ATP to maintain the tight junctions. Tight junctions maintenance is actually what costs the epithelial cells most of their energy. And so when there is a leakage of the tight junctions, that means leaking inside out towards the intestinal lumen, but also leaking outside in, which means that there's compounds that are present in the intestinal lumen that pass in between the epithelial cells, which they should not do. In case of tight junctions being closed, that should not happen. But when this happens, there's fragments of the microbes that are present in the intestinal lumen that come into contact with the basolateral side of the epithelial cells, where you have the toll-like receptors that sense the presence of bacteria. And for our body, the presence of bacteria in between the epithelial cells is a sign of danger. And this sign of danger will induce the cascade of NF-kappa B activation, inflammatory response. Consequence of that is a vicious circle of tight junction disruption. So tight junction disruption leads to activation of the NF-kappa B cascade, pro-inflammatory signals, activated inflammatory cells, release of pro-inflammatory signals again, and these pro-inflammatory signals in turn again will damage the tight junctions. You can measure this. There's a tool available which I think is really useful but technically very tough, and that is using Ussing chambers. You take the mucosa of the intestinal wall of the chicken, you create an electrical charge, and you measure the resistance of the mucosa, and that is a direct measure of tight junction tightness. And by using this, you can measure effects on tight junctions. And this is just a simple list of nutritional factors that have been shown in the literature to stabilize tight junctions. And to me, this is important to realize that tight junction stability is essential for the defense of the animal against a lot of pro-inflammatory signals, but also against the negative effects of heat stress on the intestinal barrier integrity. So here again, we come up with glutamine and with butyrate. And then this one, I think, is also very important. Was mentioned already this morning. Leaking from the intestinal lumen in between the epithelial cells will result in lipopolysaccharide from gram-negative bacteria, which is a very stable molecular complex, leaking in between the epithelial cells, entering the bloodstream, and then you get endotoxemia. Fortunately, the chicken is very resistant to endotoxemia. It will not die from endotoxemia, but the endotoxemia causes an inflammatory status. And so that adds to the inflammatory signals which were there already. And so what happens then is, in case of leakage, not only LPS, lipopolysaccharide, will pass through, but also fragments of bacteria and intact bacteria can pass in between the epithelial cells and enter the bloodstream. We call this bacterial translocation. Bacteria, when they enter the bloodstream, blood from the intestine goes directly to the liver through the portal vein. And when it arrives in the liver, these bacteria ending up in the liver, they normally should be eliminated by the Kupffer cells, the macrophages of the liver, but because of the increased bacteremia, increased bacterial translocation, there are areas in this liver where the bacteria are maintained and where they cause necrosis. And the white spots, which you can see in the slaughterhouse, when you go in the slaughterhouse, you can see these white spots on the liver of the chicken. That's actually necrotic foci due to bacterial translocation. So to me, it's very important that nutritionists should go to the slaughterhouse. They should not stay behind their computer. Go in the slaughterhouse, look at the livers of the chickens. You will immediately know when there's a problem with leakage of the intestinal wall. And then the other one, I think, which is important, is that not all of the bacteria are eliminated from the bloodstream in the liver, so they start circulating. And it's fascinating to see, you know, at one point we did an experiment in which we injected A. salmonella bacteria intravenously to broilers, injected in the bloodstream. And then we took blood samples at different time intervals. And we continued up to 14 days. And after 14 days, we could still isolate salmonella from the bloodstream, which means that the broiler actually tolerates bacterial translocation, tolerates bacteremia. And that means that bacteria that are in the bloodstream, they can end up in the bone marrow, especially in the bone marrow of the femoral head. And then you get this femoral head necrosis. And you can see this. When you open up the bird and you dislocate the hip joint, the femoral head will break. And that's due to bacterial translocation. Histologically, you can find colonies of bacteria in this femoral head. So, what is then the effect on the intestinal microbiota? Because Pierre-André asked me explicitly to comment on this, and there is surprisingly little information in the literature. I found one paper which I think is very fascinating. Don't look at this because it's not very clear, but the take-home message is very simple. The pink here is firmicutes. So under thermoneutral conditions, in the ceca of broilers, you have a dominant population of firmicutes. Under heat stress conditions, there is a shift. Less firmicutes, more lactobacilli. And then people say, oh, lactobacilli is good for you. Here are the firmicutes. Here are the proteobacteria. The lactobacilli are the ones that produce lactate. And this lactate needs to be further converted into propionate and butyrate. If that is not the case, you get accumulation of lactate. And that is very harmful. So if you have too many lactate producers, it can be a problem. And indeed, I came across this study where people were looking at heat stress conditions, and they were measuring serum D-lactic acid. That's lactic acid produced by the microbes, because the host produces L-lactic acid. If you find D-lactic acid in serum, it means that there is an excessively high production of lactate in the ceca that spills over into the bloodstream. So it means that there's overgrowth of lactic acid bacteria and not enough firmicutes to convert the lactic acid into butyrate and propionate. So that's what happens under heat stress conditions. Too much D-lactic acid, which may even cause lactic acidosis. And so D-lactate is no problem as long as it is converted into butyrate and propionate. And it will fuel the butyrate production through the Clostridium cluster 14A strains, which can convert D-lactate into butyrate. But when there's excess lactate, then the pH drops, then the firmicutes die off, because at a low pH in the ceca, firmicutes cannot survive, they die off, and there's less butyrate production. And that results in inflammation. So if I had time, Mr. Chairman, for a couple more minutes, I would just like to, there's different ways in which we can stimulate butyrate production in the ceca, which is important for the stability of the tight junctions. Butyrate reinforces the tight junctions. You can add it to the feed, or you can enhance the endogenous production. And I would like to comment on a study which we did and which was published actually last month in Poultry Science. What we did is we took wheat, soaked it with water, either incubated it or not with a xylanase complex, and then took the water-soluble fraction, so the fraction of what, the xylans that have become soluble, and then we could show that actually the treatment with the enzyme shifted the degree of polymerization to a band which is like between 15 and 30, and this was then used. We used this as an additive. You know, the extract from the wheat was then added to a feed formula, which already contained a lot of wheat. So it was a wheat soy-based formula. And we added only 0.1% of this fraction, which was like obtained from wheat being treated with enzyme in vitro, you know, in the lab, then added at 0.1% to the feed. And we could see that this actually stimulated feed intake during the startup period. And as we have heard today, feed intake is an issue under heat stress conditions. And this resulted in a significantly better body weight gain. So this wheat extract at just 0.1% significantly increased body weight gain and feed intake in this startup period. And we could also show that by simply looking at the T lymphocyte infiltration as a parameter for inflammation, we could show that there was a significant reduction in the inflammatory response at the level of the intestinal mucosa. And on top of that, to me, the most fascinating aspect is that, you know, the L cells, these are endocrine cells producing glucagon-like peptide 1 and glucagon-like peptide 2, which we heard already today, are controlling feed intake and are controlling villus length in the small intestine. And it turned out that by adding this just simply 0.1% to the feed, we could significantly increase the number of these endocrine cells. These are cells that are sitting in between the epithelial cells, and they produce this glucagon-like peptide 1 and glucagon-like peptide 2, and you increase their density. So this explains the mode of action actually of this in this study. Then looking at the microbiota, we could show that there was a significant increase in Lachnospiraceae and Ruminococcaceae, which means that we restore the firmicutes, the butyrate producers. And we could also show that there was an effect on short-chain fatty acids, especially butyric acid and acetic acid production. So to conclude, I think the most important aspect to take into account when we talk about heat stress is that this is an oxidative stress. It's an oxidative stress, so it's a pro-inflammatory stress. And so what you see is that you lose butyrate-producing firmicutes. And that's the key problem under heat stress conditions. And we know that butyrate is so important with respect to its anti-inflammatory and antioxidative activity. So you can protect against heat stress using different tools. Oops, sorry. Using different tools, but I just illustrated one of them. And I think we won't make it with just one tool. I think the future is to go for the right combination, keeping in mind that, you know, this shift in the microbiota is very important under heat stress conditions. Thank you very much.
Thank you, Richard. Really saved a lot of time. Thank you very much. So the questions, do you want to read it here and have a look at it?
So is propionate production a parameter as important as butyrate production? Propionate and butyrate both are energy sources for the epithelial cells in the lower intestinal tract. The epithelial cells of the upper intestinal tract don't use propionate or butyrate, they use glutamine. So we've seen in several presentations today how important glutamine is under heat stress conditions. And this may be part of the explanation, because glutamine is the energy source for the epithelial cells of the duodenum and the jejunum. If you compare propionate to butyrate, both are energy sources for the epithelial cells of the lower intestinal tract, but on top of being an energy source, butyrate also is a histone deacetylase inhibitor, which means that it shifts the expression of the genes in the epithelial cells towards an anti-inflammatory phenotype. Should I take the next one also?
Yeah, thank you. Yeah, next one.
This means giving lactobacillus as probiotic could increase lactate and then decrease butyrate. I didn't say that. I said that excessive lactate producing by bifidobacteria or lactobacilli can be a problem. When you use lactobacilli as a probiotic, what you use is actually, should we say, 10 to the 9th, 10 to the 8th, not more than that. You're talking about numbers that are like way below the numbers that are naturally present there. And then taking into account the lactobacilli, they don't produce spores. So 95 to 99% of them will die passing through the proventriculus, passing through the upper part of the intestinal tract. So what you add in terms of numbers will not cause excessive lactate production. I'm not saying that it will not have an effect. I'm only saying that it will not cause excessive lactate production.
Thank you. The last one is a really good concluding question, sir.
Feed intake is decreased with heat stress. Is there evidence of the role of gut microbiota in feed intake? The answer is definitely yes, because the L cells, which I was talking about, they have 2 receptors for butyrate, which means that they are influenced by the firmicutes that are present there. So if your numbers of firmicutes reduce, there's less butyrate signal going to the L cells. And that is probably one of the mechanisms by which, because L cells produce glucagon-like peptide 1 and glucagon-like peptide 2, and glucagon-like peptide 1 is very decisive for feed intake. But I'm not telling that this is the whole explanation. This is just, tip of the iceberg, probably.
Thank you very much. Please join me. Thank you.